The overall goal of the following experiment is to use high-throughput image analysis software to measure the size of three-dimensional tumor spheroids imaged with bright field microscopy. This is achieved by first arranging all the images with 3D tumor spheroids into the correct directory structure and file names for the spheroid sizer program. As a second step, Spheroid Sizer automatically computes the boundary of the spheroid based on the active contour algorithm and measures the size of the spheroid. Next, a well-designed quality control sweep is made to ensure optimal results for every image. After analyzing the resultant data, the growth of the 3D spheroids under experimental treatment can be elucidated.
The main advantage of this computer program is that it integrates a powerful automated image analysis algorithm and a quality control workflow to support high-throughput image analysis.
The software tolerates uneven or noisy image background. It reduces labor remarkably and speeds up the analysis process.
Implementing the software is beneficial for 3D tumor spheroids to become a routine in vitro model for drug screens in both industry and academia.
Spheroid Sizer requires MATLAB with the signal processing and image processing toolboxes. An optional toolbox to use is parallel processing. Download Spheroid Sizer from the Rutgers server and unzip it to an installation directory, which will be needed in subsequent steps. Now prepare your images. First, determine the image resolution of the imaging system.